All right, so once again, I'm Ed Simpson. I'm your TA, and I'm going to be talking about RNA splicing and transcriptome assembly. So I guess the first question would be, what is the transcriptome, and why is it different than exon sequencing, or what is the specifics that we're actually talking about here? So the transcriptome is just a set of RNAs that a cell produces that define the biological processes and also determine its cellular identity. And cellular identity is a key component of this lecture. So splicing is a major contributor to what cells become. And this also includes coding and non-coding RNA. So there are some additional considerations for the analysis for that. The biggest thing, however, we have to worry about when it comes to analyzing NGS data for RNA-seq, though, is splicing, because it complicates the algorithms that we use and the errors that we have to take into account. So on the left, you see just the canonical idea of how DNA is transcribed into RNA. And then what happens afterwards is some of these exons are spliced out or included, and that generally leads to the cellular identity or different functionality. And so the human genome has approximately 20,000 genes. It's thought that splicing expands the number of functional products up to about 100,000, so about a factor of 5. And therefore, it's extremely important for us to know what's going on. But given that there are actually more exons per transcript, if you multiplied that out, than 100,000, you realize that a lot of these RNA products might not be produced, and that splicing is a controlled process. So it's important for us to look into this NGS study so that we can figure out how these things are regulated and moved around in the cell. So on the right side, you see just the classic model of the spliceosome complex and how exons or introns are spliced out. So in this case, what you see is exon B is actually being spliced out. You have the U1 and U2 proteins recognizing the splice sites from the surrounding exons. And basically, the spliceosome assembles on the area that's going to be removed, and what you end up with is a shorter transcript. So aside from splicing, we also have methylated capping. We also have poly-A tails. And these are all things that are also studied in transcriptome analysis. So on the right, you see a completed transcript with untranslated regions and these additional features. And in addition to just classic mRNA, we also have to deal with the non-coding RNA in the cell that we're not so interested in, which mostly it's ribosomal RNA and tRNA, which we don't generally sequence that much. But there's a tremendous amount of non-coded RNA that goes through regulation that we can also look at. However, this isn't exactly a one process captures everything technique. So transcriptome assembly usually is defined by the experimental considerations you perform before you get to the downstream data processing. So if you want to study microRNA, it's going to be a different experimental pipeline than if you wanted to study mRNA or anything else. So again, it's different from exome sequencing in that what we're trying to actually do here is reconstruct full transcripts, or at least understand how much of specific exons are in this data set. And I'd say another extremely important thing that often gets overlooked with transcriptome assembly is that it's also with respect to generally a specific cell population. So this can be a cell type or some other controlled population of cellular identity or treatment group. Also, we want to identify the transcripts that are there, but we also want to quantify them simultaneously. So that leads to this multi-step process that I'm going to be covering. And what can we learn from the transcriptome aside from just reconstructing what's there, knowing how much of it is there? Well, some of the things that we can take into account when we do these experiments, it could be a time-dependent response that these cells are currently producing RNA for. It could be a snapshot of a production from a specific time point. We also look at the identity and quantity, like I mentioned, but mainly the identity and quantity of functional RNA elements aside from just these transcripts. And the practical applications of this is basically as simple as just the transcriptome annotation. So with DNA-seq, you get your full set of DNA, and then you can use RNA-seq and transcriptome assembly to understand which is being transcribed. And then you can also do expression studies, EQTL analysis, poly-A length. Some of the newer things that people are working on is RNA localization and circulating RNAs. And then there's also circular RNA, which is considered to potentially be a sink for RNA binding proteins, and it can help control mass effects in the cell when it responds to stimulus. One last thing, too, I want to mention about this. So you've seen a lecture on metagenomics and recovering DNA from the environment, but metagenomics tells you everything that was there. It doesn't necessarily tell you everything that is there. If you want to know what's alive, you need to recover RNA or protein from something that's being actively produced. So when you do metagenomics on an environment, it's possible everything you're looking at has basically died. Some things it can't give us, it can't exactly tell us what the whole isoform information is unless you do whole isoform sequencing, which is generally prohibitively expensive. But we use inference techniques to understand which transcripts could be in the cell. It obviously doesn't give you protein concentration, and this is becoming more of a research focus as we get more experience at high throughput proteomics and pairing RNA-seq data with proteomic data. But it's important to note that generally protein concentration may not actually correlate with RNA. And a lot of this has to do with things like the half-life, how RNA is regulated, the time scale that you're measuring these RNA samples. And then it also doesn't tell us anything that happens after transcription. This is a basic experimental workflow for an NGS pipeline of almost any type, except the cDNA prep is specific to RNA-seq. The thing that I really wanted to point out in this particular slide are all the different QC checks. And these are all opportunities for researchers to specify the type of transcriptome study that they're going to do. And the primary thing that we need to be concerned about is whether we're using RNA depletion, mRNA capture, or size selection, and some of the advanced techniques that allow us to look at different types of RNA in the cell come from this particular step in the pipeline. So further experimental concerns that can mess with our downstream analysis. Obviously, we have some RNA quality and quantity issues. It's surprising how often this is actually the limiting factor in a study. We also have RNA structure and length bias, which can prevent us from capturing or quantifying these things accurately. Again, the half-lives can be an issue. And one thing that I want to point out too is the capture rate versus the diversity. So if you're using mRNA capture and your limiting reagent happens to be the probes or the probe concentrations, you're just going to miss some random mRNA because you don't have enough probes to pull it in if they're random probes still. And then lastly, while this generally isn't too much of a concern with bulk sequencing, it's important to note that in single-cell sequencing, conversion to cDNA can be pretty low. It can only be 5 to 25% of the mRNA that's present in a single cell. And so this comes into play for building these networks. So transcriptome assembly and transcriptome studies are also an area of NGS analysis that we use some specialized methods that you may have heard about but don't fully understand yet. So I think some of these ideas have been talked about in earlier classes, in earlier lectures. But the idea here is that basically strand specificity of RNA is extremely important for a full transcriptome study, because we can have, as you see on the top, an exon that's used in 2 different transcripts. One may be transcribed going from the left to the right, the other from the right to the left. And although you wouldn't think this is so common, what we see quite often are exons nested inside of transcripts that go different directions, and also transcripts that share directionality and maybe even promoters. So considering that, one technique we use is strand-specific sequencing if it's going to be a thorough study. And basically all that happens here is you take the original strand, you make the reverse complement, and then you denature and destroy the original strand. From here you can label a complementary strand with UTPs. And really you're only doing this so that you can attach the ligated adapters. After those adapters are attached, you destroy that labeled strand. So you're just left with the reverse complement. And you know that this is the opposite of what you pulled down. Another advanced method that's commonly used in transcriptome studying and RNA sequencing in general is duplex-specific nuclease methods for extracting specific sizes of your inserts. So we know in NGS that it usually starts with fragmentation of a nucleic acid. And then ideally, we want many of these fragments to be approximately the same length. This is more important for transcriptome studies because the insert length between 2 paired reads, which is the nucleotide between the left end and the right end of a fragment that doesn't get sequenced, it gives us a lot of information about where these reads will actually land when we go back and reconstruct transcripts and do the alignments. So in order to have more control over this number and make sure that the statistics in our algorithms perform better, we can select the size of these fragments. And one method is by denaturing them and then allowing these strands to re-anneal. The smaller ones will anneal quicker, and a strand-specific or duplex-specific nuclease will then chew up the ones that have re-annealed, leaving some of the longer single-stranded copies available to anneal later. So if we go through this a couple of times, we can get highly specific with the length of time we give these strands to anneal and the temperature that they anneal at, and we can get a relatively uniform set of insert lengths. All right, so aside from the experimental issues, there's of course computational ones as well. So from bioinformatics pipelines, I just wanted to kind of clarify the process that I'm going to be talking about today. We start with a RNA-seq data file, right? And that has a bunch of reads in it that we haven't aligned yet. And we have 2 options. You can either use a reference that you already have from a previous alignment or from the Human Genome Project to find out where these reads would go. Or you can stitch them together using a de novo technique. This is still a type of alignment. So after you have the reads from your experiment aligned, there's another step, which is the discovery and quantification of the transcriptome. And this is also reference-based versus de novo. So you start by using a reference or not to build your RNA-seq file. And then you use a reference or not to find and quantify these transcripts. And as you can imagine, if you have all of these options available, a lot of times you want to use multiple techniques, right? So if you can use a reference and you can find de novo transcripts for your experiment, that would be ideal. And most of the considerations would be about which one to trust. Aside from that, we're still dealing with this short read problem, which is that short reads can be aligned to the same position, but you don't know which isoform it corresponds to. So this is a major issue for splicing analysis, which I will talk about later. But this is what's known as increased uncertainty, or the uncertainty principle, when we get to splicing analysis. Another issue is obviously RNA might not match DNA, and this could be for any number of reasons. It could be an error, or it could be RNA editing, which we found out is actually a major factor. And there are specific enzymes that locate motifs and determine which bases they're going to edit. And it is reproducible, and it's something that can screw with how we align these reads. We also have what we call RNA artifacts, or just basically things that we don't know what they do, and they're just short pieces of RNA, and they can mess with the reads that you get out at the end of the sequencing. Aside from that, I mean, it's the same problems as some of the DNA issues. You could have multiple locations where a single read aligns. You can have degradation of your sample. For transcriptome alignment, as I mentioned, there's two major schools of alignment theory. So you have the reference-based, and then you have the de novo alignments. Many of these will share the same algorithms that were used in DNA-seq alignment. So I'm going to specifically talk about the differences, but you should know that when it actually comes to scoring a read and placing it in a spot, it's basically the same thing that BWA or Bowtie 2 would use. But these techniques have been augmented to work with RNA-seq data, specifically to deal with reads that are crossing junction sites. All right, so we have the reference-based assembly, and the pipeline for this would be to align your sequence reads, and then to reassemble transcripts afterwards using a reference-based transcript assembly method. And then you have the de novo transcript assembly, which can serve as a reference as well downstream. So for the reference-based alignment, the original technique was simply to use a DNA-seq aligner, and then to get a list of junction sites that could potentially match your reads, and to just search that list as an index and try to put a read that had pieces split between two different exons in the position where you thought it would go. There's also some more modern techniques we have, which are just splice-aware aligners, which can sort of do this on the fly. They can build indexes and notice anomalies when they try to place these reads that minimizes the amount of time and the errors that they make while they're actively doing this alignment. So a couple of these techniques you've already been introduced to, and I don't want to spend too much time, so I'm going to try to go through this pretty quickly. But Dr. Lee already talked about the star aligner, so the biggest thing I think you guys should be aware of for this is it was originally designed for the ENCODE project. It was 50 times faster than any existing technique at the time. And mainly its biggest advantage was its speed, because they didn't have the time to wait around for these alignments to be done when they're dealing with this kind of data. But a trade-off for that is you load a tremendous amount into memory. So the way that it works is basically you align the left end of the read or the right end of the read. Generally, this is done simultaneously. For paired reads, you match as much of it as you can. As soon as you hit a mismatch, you take that and extend it somewhere else in the genome. You consider the length of the intron, and you also consider the length of the insert between these paired reads so that you have an approximate location in the genome where you can stitch the second end. That's actually done with the suffix array, by the way, which you guys have learned about and actually had on your exam. So step 2 is just kind of resolving both of these reads from a pair so that the distance between the two ends of the split reads makes sense. But again, the biggest, I'd say, factor in this is the insert length distribution, which is the distance between the two ends of the reads. So this figure is just an example of how star compares to a few other popular aligners at the time. And the main thing you can see is a massive advantage in mapping speed, or specifically the number of read pairs per hour that you can put on a genome with star. And this extends no matter how many threads you use. So that was by far the coolest thing about star. A more modern alignment technique that we have now is high-sat2. So many of you have heard of Top Hat or Top Hat 2, and Dr. Leo briefly introduced Top Hat. This is done by Steven Salzberg. This is the same person that actually did Top Hat and cufflinks. And even he recently published a paper, I want to say it was maybe 3 months ago, where he told everyone, please stop using Top Hat. High-sat2 is better. Don't use my old tool. This is the thing you're supposed to be doing. And you can imagine that's an issue. A lot of people will pick up tools that they see in papers, especially in big papers like TCGA or CCLE. The issue with these large consortium papers is that they use the technique they had at the time, and when they make new data available for these consortiums, they maintain the same tool. So even though that tool has been out of date for going on 5 years now, they continue to re-release new data using an old tool because that was the pipeline they developed and they want to be consistent. And some of the fallout from that is we're stuck using outdated things. So always keep in mind that there could be something better out there. The biggest advantage, though, for high-sat2 compared to some of these older techniques is low RAM and that it's actually even faster. So it kind of solves the RAM issue from star, and it still brings the speed. Aside from those general considerations, the way it accomplishes this is by using two indexes. It uses a global index, which we're familiar with from an aligner like star, and it uses a local index. The global index is a lot less specific, and the local index is more specific. So you sort of just ballpark where you think a read is going to go, and then you pull a secondary reference and match it there faster. And the biggest thing from this comparison that I want to draw your attention to, while Top Hat 2, you can see still performs well, high-sat2 over here is the best, and it's approximately similar to star, what we call two-pass mapping. A lot of people still use star and high-sat, and I think they're both very viable. But the biggest advantage you can see from high-sat is the memory. It uses about as much memory as we would have on half of a modern computer. So this is something you could run on a desktop. However, you still need memory to build the index, and that can mess with your pipeline if you don't have access to a machine that can do that. Okay, so that kind of covers the reference-based alignment. I want to get into de novo assembly, and again, I'm not going to spend too much time on this because Dr. Lee already talked about this. But the most commonly known technique is Trinity, and it's sort of established basic pipeline for other tools to follow. And from here on out, you're going to notice a lot of tools where I use the words like Kamer and De Bruijn graphs because these techniques are all like relatively universal. But the way they implement them and the considerations around that are what kind of separates them, what makes them different tools. But essentially, Trinity uses a three-step process where each step builds upon information from the previous one. You start by making a list of Kamers. You remove erroneous Kamers. And then you extend in both directions Kamers which overlap based on the frequency. So you start with the most frequent Kamer, and then you extend in both directions, and you get a transcript. And then you drop down a level of frequency, you pick another Kamer, and then you extend both directions, you get another transcript. That's step one. Step two is that you're going to merge these different transcripts that you've identified into a De Bruijn graph, basically. You break it up into these smaller word sizes. And really all you're trying to do here is merge transcripts that you think are actually the same and to reduce the size of the reference that you're going to use later. So finally, when you actually reconstruct the transcripts that you think are in your sample, you're going to pull these reads and stick them onto Kamers that they match. And then you're going to try to validate that by, again, looking at the distance from where this Kamer is to another Kamer, say, farther up in the graph. and trying to, I guess, actualize that with the distance from the insert length distribution between these two reads. So once again, you're bringing in information from how far apart the paired reads are from each other. And this is a key component to all transcriptome assembly. All right, so now we have some type of reference that we can use to search for more transcripts and to count how much of each transcript is there. So I want to get into the techniques about doing those specific steps in this pipeline. So one of the common tools is RICM, but more, I guess, classic is cufflinks and scriptures. So these two, they sort of go hand in hand, and they were the forerunners of a lot of techniques that we use today for full transcriptome assembly. And specifically, that's whole isoform quantification. So cufflinks is the widest tool used for isoform quantification. This is because it was released basically with Top Hat 2 as a workflow. All these tools were released by the same laboratory with the explicit design and already prepared pipeline. So all researchers had to do is just pick this up and get running. It includes multiple tools for downstream analysis, and this includes things like merging cufflinks results from multiple samples so that you have one large transcriptome. If you're analyzing a bunch of samples that you think might have unique isoforms, and then this is a good option. The way cufflinks works is pretty simple. The first step is that you want to identify reads that could not possibly come from the same isoform. So here we have a couple highlighted. The dotted lines indicate a split read that spans an intron. So you can imagine that a read that spans the yellow isoform has no evidence for the exon in the center of this blue read here. So it could not come from the same transcript. The next thing is that you would build a graph from the surrounding reads to understand which of these paths could be responsible for this transcript. So this is also a relatively simple process. You just simply overlap the ends. And what I want you to pay attention to here is the thicker boxes are the reads, and the thinner line is the insert that is not being sequenced, just so you're aware. After you have this graph, you go ahead and you build the minimum number of paths that explain these transcriptional states. So this word minimum is extremely important here because cufflinks isn't designed to exhaustively search the transcriptome for novel transcripts. It's designed to find transcripts with evidence and really to maximize their size. So after we understand what we think is in the sample, now we want to figure out how much of it is actually there. So the way that we do that is we have our 3 different transcripts, and they each are different lengths, different coding sequence lengths. And we have reads that support each of them. And so if we want to count how much of these could potentially be there, we have to assign them and specifically, again, consider the length of the insert between these 2 reads. So if you look at this purple read, you'll see that it was assigned to the blue transcript originally. And the reason why this happened is because this insert length is pretty large, but it's still a lot smaller than if this had been assigned to a pink transcript. So it would make more sense to assign it to the blue transcript. And after you're done sort of calculating what the lengths of these transcripts are, you get the log likelihood that reads are assigned to one transcript or another. So you take the sum of all the transcripts in the transcriptome, and each of them has a log likelihood. And then you put reads into them, and you calculate the new log likelihood in an iterative procedure until you optimize this equation. And finally, what you end up with is your abundance. So this is based on the probability that you think these reads should be binned into these different transcripts, modified by the abundance. Scripture is another tool that's reference-based for reconstructing transcripts. This tool is a little different in that when you determine what a transcript is, you have a sliding window score, and this also considers constitutive reads. But before this point, you generate transcripts purely based on junction reads. So the sliding window takes into account the density on exons that can modify the probability that a read goes to one transcript or another. The biggest difference that I want to call attention to with Scripture is that you consider all possible paths in the transcript graph. You assign a statistical significance to each of these, and you can filter this significance so that you minimize the number of transcripts that you look for. But the real strength is that using Scripture, you can identify more transcripts generally than you can with cufflinks. It's a way to fully enumerate the possibilities of transcripts given your RNA-seq data, and cufflinks is a way to maximize the specificity. So this is a question I think Dr. Liu likes to ask. So you should probably know the differences between these two tools. So the next topic I'm going to briefly talk about is this alignment-free transcriptome analysis. So this is a relatively new process. The original publication, which kind of started this trend, was Sailfish. That's not exactly new, but since the developers of the previous tools like cufflinks have started their own laboratories and moved on from their postdocs, they're more interested in this technique because it's a lot faster. It requires a lot less time and memory. So one of the things you'll learn too in these particular sets of algorithms are that they do have a lot in common, as I mentioned before, with Kamers and De Bruijn graphs. And sometimes it's splitting hairs, but the difference between accidentally getting something in common and purposefully stealing something has been debated in the field as well. And so there's a big debate with two tools specifically that I'm showing in this particular slide. It's between Callisto and Salmon. And so Salmon was developed by a group that was pretty famous, and people had been talking about this new tool for alignment-free assembly for a while. And people were aware that Salmon was going to be coming out. And Callisto was developed independently. And it seemed to be incredibly fast while it was being published. But they were released for preprint. And what happened was the results from Salmon, if you want to look at the plot on the left, for differential expression of transcripts, they're virtually identical to Callisto. After these papers were published, this drew some attention. And the creator of Callisto, Leo Pator, went back and plotted the different versions of his particular tool against Salmon and against Sailfish. And what he saw was that basically as Salmon was being developed, it became less like Salmon and it became more like Callisto. And he tracked back the individual code snippets that had been updated, including blocks that appeared directly copied from his particular pipeline, which was a bit alarming. This continues to be a debated topic, and it seems like Salmon is unique enough that no one in the field has caused some sort of a riot over this or been fired. But the only real major difference between Callisto and Salmon is that Salmon uses GC correction and Callisto does not. And so the debate goes on about whether the plots were skewed to make them look more different and who's really responsible for the main idea behind the analysis pipeline. So just to introduce this set of algorithms without getting into all of the different types and varieties, I'm going to talk about the oldest, which is Sailfish. It's Kamer-based and it has that expectation maximization feature Once again, what you're doing is you're taking the transcriptome, actually, and breaking that into k-mers. So that means that you need a reference of transcripts before you look at your read data so that you can build this database to not necessarily align against, but to bin your reads into. So you take a list of these transcripts, break them up into k-mers. You count approximately how many k-mers are associated with a particular transcript. And that allows you to assign your reads to these different bins. And then once you assign your reads to a bin, you go back and you calculate the probability of a read being assigned to this based on the number of k-mers from this read that match that particular bin. And again, it's one of these iterative algorithms where you have the likelihood or the probability, you have the actual number that you've counted, and you use them to optimize each other until you converge. So after we've done our RNA-seq analysis, we have this set of aligned transcripts. And in general, how do we know what the quality of our RNA-seq experiment really is, especially if we've never sequenced an organism before and we don't know what to expect in terms of the number of contigs or anything like that? RNA-seq quality is a little bit more challenging to understand than DNA because we don't know how much of each transcript we should expect to see by default. We don't know like the default state of each cell, especially if you're doing bulk sequencing. There's many default states in one sample. But we can look at the read counts to see how much corresponds to each transcript. We know that multiple copies of each transcript exist. And we know that there's going to be duplicates, which means that we don't want to remove duplicated reads. So that's generally a common practice for calling variants in DNA-seq. In RNA-seq, duplicates are actually what you use to infer the expression of something. So if you remove those, you can't do the rest of your analysis. But the question remains, how duplicated should one transcript really be? We still get a Phred score, so we still know approximately how accurate our reads are. But the biggest quality indication we generally look at is the fraction of reads that map to a particular genome. Or if we're doing a de novo assembly, you really want to look at how many reads you can track back to the individual transcripts that you're trying to understand. And we also look at the percent of reads that map to the intron or the intergenic region. So I would say that this is a key factor in the quality of RNA-seq analysis because generally you don't expect reads to be in the intron or the intergenic region if you're sequencing mRNA. Most of that should have already been spliced out. If you see reads that fall into introns, it could mean that your RNA is degraded, or it could mean that it's mostly artifacts, and it's likely poor quality. One other thing I didn't put on this slide is the ratio of forward strand to reverse strand reads. So many library preps involve capturing only minus strands. So if you see a poor ratio where you have a significantly large amount of plus strand reads in a library that should only have minus strand reads, it's similar to the off-target issue of landing in introns. All right. So splicing analysis from transcript isoforms is another problem when it comes to this transcriptome assembly. And what you're looking at here is a graphic of a cell that's produced multiple transcripts. And then you have exons, the introns have been pulled out. And this is what the data looks like when you generally just sequence the individual reads. But when you pull these back and align them to a reference, or if you reconstruct a transcriptome of an organism, the data actually looks like this. So reads will land on a piece of an exon, and then you'll see these long stretches, which is coded by ends on the actual read in the BAM file. And then you'll see this piece of an exon landing with the other part of the read. So the biggest thing that you can look for when it comes to alternative splicing just from your raw data is the mixture that you have of reads that align to different transcripts. So if you look at these two reads on the bottom here, what you're seeing is one read that corresponds to these two exons and another read which suggests that this exon is not included in a particular transcript being produced by this cell. A lot of the mathematical models that we use to reconstruct the splicing information is based on the proportions of those reads, specifically junction reads. So when it comes to splicing in general, it's important to understand the regulatory context that this happens in. So here we have a schematic of different RNA binding proteins, specifically splicing factors. And we can see that in the various transcripts that are being produced, you can have multiple splicing factors producing the same transcript. You can have one or two splicing factors being necessary, where in other cases maybe more would be necessary. There's also been discussion of not only the context, but the concert action of splicing factors, which is if you have one splicing factor binding next to another splicing factor, it could modulate the activity of them together. So it could either cancel out what the previous splicing factor is trying to do, or it could augment it and make it more effective at splicing a particular exon. And then, like I mentioned, the concert, the context is basically, if you have a splicing factor in front of an exon, it may enhance splicing. And if you have a different splicing factor in front of that same exon, it may inhibit splicing. So it's all a very complicated network, and these things all work together to make it particularly frustrating to understand. So to get into how we look at exactly pulling apart this network, we need to know, first of all, the types of events that we're going to be dealing with. So there are approximately 5 common types of events. You have the standard skipped exon, and then you have alternative 3 and 5 prime sites, and you have mutually exclusive exons, which are basically never in a transcript together. And then you have intron retention. When we quantify the different types of spliced events, really what we're looking for are the approximate number of transcripts that we think have an exon versus the number of transcripts that don't have an exon. So this is an exon-centric approach, meaning that we don't necessarily tie these back to their individual transcripts or the combinations of exons that are present in one specific transcript. So if all we can understand is the percentage, we can calculate that relatively easily in a simple equation just using the junction reads. This ratio of included exons to excluded exons, or included exons for transcripts versus the number of transcripts that don't have it, is known as the percent spliced in value. So down here, we see that there are reads that span particular junctions, and we have what are known as inclusion reads. So this is the reads that span the junction that is part of the transcript, as well as the reads on that exon. And then we have exclusion reads. So these are basically only the reads that say that this exon isn't there, and there's not very many of those. And then we also have constitutive reads, which tell us essentially nothing. It could belong to either of 2 or 10 isoforms, which we're not really certain on. If we want to get a basic understanding of the percent of transcripts that have this exon, all we have to do is take the junction reads for the inclusion site. So in this case, there's 6. We've got 3 here. We've got 3 here. And then we actually divide that by the total number of reads that support or refute that event, but only using the junction reads. So if you look, there's 2 junctions next to this exon of interest. So that means that there's 2 possibilities for us to identify reads that support it. However, if we're looking at the number of reads that say that it's not there, there's only 1 junction site. So in the denominator, we actually multiply that by 2 so that it normalizes the probability of us seeing reads that are associated with inclusion or exclusion. So that's why we get this formula over here. And while in this case it looks like there are many more reads that support the inclusion isoform, it's actually a 50% chance of observing both. So this again is one of the things that you may be asked to do on a test to calculate the PSI value for a specific exon. So there are, again, a lot of challenges dealing with splicing information. We've got events or exons that are different sizes, and you guys have probably heard of microexons by now, which are tiny exons that play a major role in neurobiology, and we're only now gaining the tools that we need to understand those small pieces of exons and how they can dramatically change where transcripts go. But there's another assumption that we make a lot of times when we analyze these events, which is that you have an equal probability of putting these reads at any position in the event. But actually we know this isn't the case, because just doing an alignment means that a base that's next to an intron could be flagged as an error. And then we split that read to look for its next location. And if we don't identify that location correctly again, then it could also be identified as an error, or the read may not even map. And so we're looking at less reads surrounding the areas we're really interested in. Something that helps to ameliorate this fact is using longer reads, which can dramatically improve splicing analysis results. But again, accounting for the insert length distribution can help us figure where these reads should go. So again, I wanted to bring up the uncertainty principle, which is that we have many of these reads that could align to one isoform, they could align to 10 isoforms. We have absolutely no idea. And then, of course, technical norms. So the gold standard in this individual exon quantification is MISO, or the Mixture of Isoforms tool. It's been proven to be highly accurate. And big advantage is that it uses constitutive and junction reads. And I'll talk about how it does that in a minute. But essentially, it doesn't give us the exact number that we need, which is how many exons we can expect, how many isoforms we can expect to have this exon, and how many we will not. What it does is it lets us know how wrong we think we will be. So it gives us an estimate of the error around a specific event, which is really important and can later be used for filtering when we want to tie these back to functional studies. This paper also introduced the idea of normalizing read alignments based on the length of the alternatively spliced events that you're trying to align to. And this paradigm has been kept in the splicing field for a while now. Unfortunately, it's not particularly fast, so that's its biggest downside. So basically, the way that it works is that we sample reads that correspond to one particular isoform, which is either the included or the excluded isoform for an event. And you can just picture a standard skipped exon event, which would be this. What we do is we pull junction reads and then sample constitutive reads around the exons that are next to this. And we calculate the PSI. And then we repeat this process 2,700 times. And at the end of the day, you still don't know the real answer, like I mentioned before, but you get a confidence interval. And this confidence interval can tell you some very valuable information about this exon, more than just how wrong you are even. It can tell you about how important this exon is to the organism in general. So if you look at the right side, what you'll see here is a relatively normal distribution with a percent spliced in value for an exon around 0.5. And you'll probably infer from this that this exon is maybe important 50% of the time. And then if you look at this bottom event, you'll see that the distribution is much narrower. And generally, it's only included approximately 18% of the time. So this is another filtering criteria to do something interesting with this particular event. Unfortunately, there's some dangers with analyzing splicing information this way. The fewer reads you have that support an event, the narrower this distribution is going to be, because you're going to be drawing the same reads every time, which means that your PSI is essentially going to be identical. And it can give you somewhat false confidence, but MISO considers a number of reads as well that it's using to draw these conclusions. MISO doesn't have a direct way to compare large numbers of samples. However, you can compare one sample to another using a Bayesian information algorithm that MISO provides as well. But the biggest improvement that you can see MISO allowed us to do is to actually narrow the amount of uncertainty that we had around an event. So while previously we could calculate a PSI of 0.5, the accuracy for this event, if we didn't use constitutive reads, could vary wildly, and in many studies may not have even been considered. But with MISO, filtering this and then comparing the distributions from each of these samples to each other, we get a much more accurate assessment of what the estimated percent spliced in actually is. And you can see that in this particular plot, the more reads we have that cover a particular transcript, the more accuracy we can expect. And regardless, MISO gives us a big advantage, but it's specifically useful in the case where you do not have a lot of reads. So after we have a PSI value for a particular event and a confidence that tells us what we think is potentially going on, we can plot this raw data. And this is a sashimi plot, which if you study any splicing, you'll likely see in a lot of papers. And this pulls directly from the read alignments. So on the bottom, what you see is essentially the number of reads that support the exon being excluded. And on the top, you have the two junctions, which would add up to the number of reads total that support inclusion. So if we wanted to compare individual events across samples, the best tool currently is this tool called RMATS. So this uses the same statistical model in a sense that MISO does, in that it normalizes the number of reads that we get based on the length of a particular event. And it reports results for both junction and constitutive reads, and it will also report results for just junction reads. So it's not particularly fast and efficient, but it has been updated so that it does work a lot quicker. And it's actually been deployed on Docker, so you don't have to go through all of these ridiculous steps just to get a package to load, which is nice. But even more interesting is that its statistical model is available as a solo package. So if you don't want to deal with the lag time of the original tool, you can count the reads yourself, but this takes some expertise and experience. As far as some limitations go, the biggest one is that it requires a uniform read length. So generally in a sequencing experiment, you would expect to see about over 85 to 95% of your reads being your target read length. So if you're doing a 75 base pair sequencing experiment, you expect 80 to 95%, 75 base pair reads. If the quality of your experiment was not so great, you may need to pull reads from other lengths. So it could be 74 base pairs, 73. You may end up going down to 72 base pairs before you can get about 85% of your reads recovered from an experiment. Unfortunately, RMATS, you have to use all of the exact same read length size. So you would lose all of those reads, and you may only be recouping about 65 to 70% of your reads to do splicing analysis, which can dramatically hurt your power. But it does identify low coverage events. Unfortunately, It also flags them as significant, so you need to do some raw filtering. And the way that works is you'll see like a p-value and an FDR, and you simply have to look at the raw counts around a particular exon to make sure that you're using something that really makes sense. Some advantage as well as a disadvantage is that it automatically searches for unannotated events, and you can request that it search for novel events. So it's widely known that much of the annotation that we have in GTF files available to us for even the human transcriptome is incomplete. And individual RNA sequencing experiments may indicate that transcripts exist that have never been documented before. If you analyze multiple samples with RMATs, it will pool together all of these novel transcripts and quantify them. But if you're trying to do a particularly controlled experiment, or you only want to use something where you have a lot of evidence for, this is actually a weakness because it ends up diluting the statistical power of the test. And now you need to pass a significant higher threshold in order for the events that you're interested in to show up. There's no easy way to turn this off, but with some tricks you might be able to. The last thing about RMATs is that it's not great for a large number of samples. So this seems to be ideal for 3 to 5 samples. Anywhere up to 12 or even 20 samples maybe is doable per group, 20 samples per group with RMATs. If you get above this, it starts to get untenable, both in resources and in time. And so that's where some advanced methods for splicing analysis need to be used in order to get an answer. So again, I want to come back to this idea of reads essentially mapping to any position with equal probability. So this is just as much of a weakness as it is for RMATs as it is for other techniques of quantification. But the nice thing about RMATs is that it accounts for variance across all of your samples, and it also accounts for variance in your specific group. So let's say you have a splicing event that becomes unregulated in one set, but is highly regulated in another set, and you compare these two. Originally, the differences between this PSI value might not be so big, but the variance and the parameters of the probabilities will be very different. RMATs may be able to detect an event like this, whereas other methods would not. So lastly, I just want to briefly mention this idea of the advanced methods I brought up. So these methods center around generalized linear models. I'm not going to get into too many specifics about how they work, and I just want to mention that basically they're possible, that you can quantify huge numbers of reads in an extremely short amount of time. So just to give you an idea, I use one of these methods to quantify about 500 splicing events, 200 per group. I was able to quantify like 100,000 events, I think, in about 10 or 15 minutes. And with RMATs, that would have likely taken me months, I think on the order of months, if not more. But once you break something down into a simple equation with read counts, it can be processed much quicker. As long as you do it the right way, it's not too much of a trade-off. All right, so we've looked at some of the transcripts. We found some exons that are interesting to us. And now we want to know what's regulating these transcripts. Why do they exist? What can we really learn about the exons that are in these isoforms? Why is it important? So just to give you an example of one of the downstream techniques you can use, a popular method that you start with is looking for motifs of RNA binding proteins that could influence the splicing of these particular exons. So here we have a skipped exon event, and actually there's 7 regions of interest around the exons and introns for this event that RNA binding proteins could be influencing the splicing. We've got this constitutive exon, and we want to look at the junction site both upstream and downstream of this, right? So we've got two pieces of sequence we want to look at there. We've got this target exon, and really the RNA binding protein could associate anywhere along this entire stretch, and also flanking its intronic positions. So usually when we do this, the flanking positions, we analyze anywhere from 150 to 300 base pairs of, but there have been associations that are generally within this range and very few that fall outside of it. So that's why we kind of limit our search space to that particular size. We use the entire target exon, but we will only use a small portion of the constitutive exons around it. Okay, so I think now you should understand which of these regions matter when we're pulling this information. We've got 7 regions. We're going to analyze each region separately, actually, rather than combining them into one sequence. We scan a list of events. In this case, we'll say that we identified 200 events, and we scan region 1 for all 200 events to look for motifs. And we do this with a tool called FEMO, which is part of the MIMI package. You can also do it with a tool called Homer, which is a more popular method now. It seems to be replacing some of the older motif scanning methods. But really all we're trying to do when we do this is count the number of times we see a motif for our whole set of events. So 200 events. Do we see this motif 10 times? Maybe we saw it twice in one event and once in another event, but we're still just going to add up the total count. And that gives us a list for each particular motif. And so now what we do is we draw events randomly from the genome. And we do this about 10,000 times. And we pick the same number of events as the number of events we have in our analysis set. So if I have 200 events that are interesting, I'm going to randomly draw 200 skipped exon events from the genome and count the same way I just counted region 1. And this would give me a distribution to the right. So you could see this is 10,000 draws randomly from the genome. This red line was the number of times, about 15, that we actually found the motif in our set of 200 events. And what this tells me is basically we saw this motif at a statistically significant separated number compared to the background that we would expect to see it randomly. And so that means that it's basically enriched. So this is what we call motif enrichment. We have more of it than what we would normally expect to see. The next step after this, we know that we see this motif more often, but getting some more meaning out of what this motif could represent for our specific splicing event can be a little tricky. So this plot is going to be a bit staggering at first, but it has a lot of strengths in it. So let me just quickly explain what's going on here. Now that we know that we have a particular motif, we have a p-value, and that's for the entire set of events. But we know that events can have more of a particular exon or less of a particular exon. And we have exons that have both been included more frequently than you would expect and included less frequently than you would expect. So we can associate a motif with increasing the amount that we see an exon, or we can associate a motif with decreasing the amount of times we see an exon. The way that we do this is, again, by counting the number of times we see it in events that have the exon compared to the number of times we see it in events that don't. And we can do a Fisher's test on this table for an association p-value. So what you see over here is actually a Fisher's p-value that says that it is significant that this event was associated with less of exon, and this event was associated with more of exon for our particular samples. So, well, that's kind of a roundabout way of explaining things. At the end of the day, what that means is if this motif corresponded to like SRSF1, and this motif corresponded to RB-FOX1. We could say that this particular motif and its associated SRSF1 protein enhance splicing in our sample, and therefore the expression of this SRSF1 protein may indicate that the splicing changes that we're observing will take place in the future. We may even be able to manipulate some of the exons in the future. So that's kind of what this table is saying. This over here is a principal component plot, and what you see plotted are the actual motifs themselves, and I've annotated some of these with the sequence logo from a class of motifs that correspond to that representative sequence. And in this case, the blue line corresponds to events that the exon has been essentially removed. And then the orange line corresponds to events where there's a new exon that's included compared to another group. And really, if you just see an association in this plot with the axis of those two lines, it can tell you essentially that a motif is associated with more exon or less exon. So the way this plot is generated is we just look at the number of times that we observe it in each group. Again, it's simply using the counts, but the difference is there's no significance associated with this plot. We have the significance on the right side that tells us it's important. And on the left side, maybe it's important, maybe it's not, but there's a group of motifs that all look highly similar. Over here, these red arrows seem to indicate these motifs actually have a lot in common. If they didn't pass the significance test, that doesn't mean that they're still not interesting, and it could be a target for functional analysis later. So that's sort of the point of doing this complicated motif analysis and association study. So after we have our associated RNA binding protein, now we can actually use the same RNA sequencing data that we used to build this transcriptome to understand if the expression is different than what you would expect compared to another group. So let's say in region 3, I identified the RNA binding protein ELAVL1. If the motif is there but the protein is not, then who cares? You know, there's no reason to even consider the RNA binding protein as being associated with splicing. But if we can go back and prove that in the group that we're interested in looking at, that this RNA binding protein has increased expression, it just makes it even more likely that it's having an influence over the alternatively spliced events that we're analyzing. So if you go ahead and you do differential expression analysis, you can then find these same RNA binding proteins, and it will be another step towards demonstrating this link, this causation. And this is all done computationally before you even get into a lab to validate these things. So the next thing that we can do computationally is say, all right, we've got our RNA binding protein and we have our exon. What does the exon actually do? You know, like, do we care that this exon is there or not there? Is it part of a protein domain? Is it part of a kinase domain? Is it a post-transcriptional modification site? Could it be a transmembrane region? You know, does it even matter if we keep it or destroy it? So the way that we can look at this, unfortunately, there's not a tremendous amount of tools that allow us to find this specific association, but there is one which Dr. Liu made very clear that he wanted me to tell you guys about because it is his tool, and as far as we know, it's the only one out there that does this kind of prediction at the moment. It's called exon impact, and it works actually very similar to the way that some of the SNV algorithms work when you try to understand if a SNP is important. So you take your region of interest and you pass it to this algorithm, and this algorithm was trained to quantify the percentage chance that exon will damage or change the structure of your protein versus the chance that it will have no effect. And the data that was used to train it specifically is related to the evolutionary conservation of this particular region. It's related to the structure of the RNA. It's related to the protein sites that could overlap this particular exon. And it's related to the existence of a post-translational modification site. So we string all of these together in a logistic equation, which gives us a probability that this is important to you. And this has already been pre-built, so all you do is you feed your event ID into this algorithm. The only downside of this particular algorithm is that we're looking at an individual exon without the context of the isoform. So now we have to report the probability that it damages transcript 1 and 2 and 3, and so now you don't really know which transcript is actually active in your cell. So the best way to try to understand which damage prediction you want to use is to either look at the predicted exon abundance, or essentially which transcript has been annotated as the predominant isoform for a particular cell or sample size, or to just find a way to combine these damage numbers and hope that it's accurate. And so lastly, I just want to mention functional validation for some of these exons and what we can do. We could use shRNA either to knock out transcripts that have an exon by targeting the exon itself with the shRNA. Unfortunately, this could favor knocking out the entire transcript and not just a single exon. So there could be other exons on this transcript you're knocking out as well, and you're changing the dosage across the board. There's CRISPR, which is seemingly a better solution, at least theoretically, but it's not often easy to do. You can still eliminate the exon entirely from the genome and see what kind of results will happen downstream after you express this transcript. And lastly, this is maybe one of the less favorite options, is splicing factor manipulation, because if you change one splicing factor, as we saw earlier, there is a very interconnected network of splicing factors that talk to each other, and you're likely changing a lot more than just the expression of a single exon. But this can be a good option if you're doing a study, for example, in cancer, where you see a particular splicing factor increases in expression dramatically in your cell group compared to another. And there are splicing factors that are associated with cancer whose increased expression can cause things like epithelial mesenchymal transition, metastatic behavior, mast cell proliferation. And that's all I have. Any questions?
Can I ask about the homework? Is that okay?
Yeah, that's fine.
I sent you an email actually about this, but I forgot to ask
Yeah,
in class.
yeah.
So for BWA, you have specified, I think it was version 0.7.15.
Right.
Is it okay to use, if